Today we'll be talking about stem cell biology in periodontal disease. Next slide, please. This video explains the role of stem cell biology in periodontal disease.
Hi, I'm Dr. Joe Nemeth. I'm a periodontist in Southfield, Michigan, and I want to talk a little bit about a possible future for dentistry, and it's regarding stem cells. There is research being done now at the University of Pennsylvania, at Tufts University, and I think other institutions around the United States, possibly around the world, on utilizing stem cells, human stem cells, to build and grow bone or revive actually dead teeth. Now, there are 5 different sources of stem cells in the mouth around teeth. There are stem cells from the ligament or that's the tissue between the tooth and the bone. There's stem cells in the pulp or in the center of the tooth where the blood supply and the nerve are. There are stem cells around a follicle or a baby tooth that's developed before it comes into the mouth. And there are stem cells at the tips of the roots of teeth. So there are a number of sources of stem cells. These institutions are trying to see what they can develop with the stem cells. I think in some cases they're actually implanting them in the bone and actually growing teeth. Well, my ideal would be to take some of these stem cells, maybe from the ligament between the tooth and the bone, and somehow be able to activate them so that we can get new bone and new attachment to the tooth that's losing bone from gum disease. So once we clear up the infection, if we can stimulate stem cell growth in that area, which can then stimulate bone growth, it would be a huge, huge advance. And I think someday it may happen. Right now, we don't really have anything that is really practical for doing that, but there's ongoing research. I'm very interested in it. A friend of mine in Florida, Dr. McCauley, is very interested in it. We're going to be doing some more work and research to try to find more about what possibilities there are. And certainly I'm going to keep you updated on it. But I know stem cells are an exciting new area of research. People are interested in them. And I just wanted to talk about them a little bit and what is so far starting to happen in dentistry. Please.
So what do we understand by stem cells? Stem cells are actually the body's raw materials. They provide new cells for the body as it grows and replaces specialized cells that are damaged or lost. They have 2 unique properties that enable them to do this. They can divide over and over again to produce new cells, and as they divide, they can change into other types of cells that make up the body. So they can divide into blood cells, brain cells, heart muscle cells, or bone cells. No other cell in the body has the natural ability to generate new cell types. Next slide, please. So what are the periodontal ligament stem cells? The periodontal ligament stem cells can differentiate into cementoblast, osteoblast, fibroblast. These are the main cell types responsible for guaranteeing tissue hemostasis of the cementum, alveolar bone, and periodontal ligament, respectively, which highlights their application in regenerative periodontology. And they can also differentiate into neurons, cardiomyocytes, endothelial cells, pancreatic islet cells, and keratocytes, which can also be used in regenerative medicine. Next slide, please.
What is stem cells biology in periodontal disease? And what do you know about regenerative periodontal therapy? So, periodontitis is an infectious and inflammatory disease that affects the immune system and destroys the periodontal tissue and teeth. Conventional dental treatments can address this issue. However, periodontal restoration is not yet possible. So, regenerative periodontal therapy rebuilds alveolar bone, gingiva, periodontal ligaments, and cementum. Stem cells, growth hormones, and extracellular matrix scaffolds can also repair injured, periodontal tissue. Mesenchymal stem cells, embryonic stem cells, and in this pluripotent stem cells have been shown to regenerate periodontal tissues. Researchers have tried to harvest stem cells from dental tissue like ligaments, gum, dental follicles, pulp, apical papilla, and exfoliated human deciduous teeth. Stem cell based tissue engineering strategies aim to restore periodontal tissue, including the periodontium, dental pulp, root, dentin, and alveolar bone. Next slide, please. The goal of stem cell therapy for periodontal disease to enhance the body's natural regenerative ability and speed up healing. Modern stem cells research focuses on tissue engineering techniques that attempt to regenerate periodontal tissue, including those that can occur in the periodontium and along the whole tooth, dental pulp, root, dentin, and alveolar bone. Next slide, please. As we said, like in the I mentioned in the previous video, that's research is still ongoing about this matter. So now we're gonna talk about as we look at this picture. This is an insuration of healthy periodontium, periodontitis, and scaffold-based regenerative approaches. On picture A, we can see a healthy periodontium, which, as we already know, is consisted of junctional epithelium, connective tissue, fibers, cementum, periodontal ligaments, and alveolar bone. On picture B, we see periodontitis with bone resorption by activated osteoclasts, formation of dental plaque calculus and inflammatory responses with an increased number of neutrophil and macrophages. On picture C, surgical procedure was performed and it involved root planning and remove calculus, necrotic cementum, and inflammatory granulation tissue. Then on picture D, as we default treatment option, guided tissue regeneration with contact inhibition membrane is frequently performed and it can be combined with filling that periodontal tissue gap with various scaffolds. Then on picture E, multi-phase scaffolds can be implanted to induce indurative regeneration of multiple periodontal tissues. On picture F, the implanted scaffolds are expected to undergo degradation as new tissues are forming.
Antimicrobial effect of scaffolds for periodontal regeneration. The major etiology of periodontitis is bacteria, for example, Porphyromonas gingivalis, Tannerella forsythia, and Treponema denticola, and host immune responses to them manifest the disease. Periodontal treatment involves removing dental plaque and calculus, but the recurrence rate of periodontitis is linked to maintenance to prevent secondary infection. So avoiding plaque deposition around the surgical site is crucial for successful surgical outcomes. That is why antimicrobial function has been incorporated in various scaffolds designed to support periodontal regeneration, as well as to minimize the potential reinfection after the initial periodontal treatment. An example of such component is chitosan, a natural seashell-derived polymer with antibacterial, antifungal, bioadhesive, and hemostatic effects. Several previous studies showed the promising effects of chitosan in bacteria causing periodontitis. For example, chitosan particles inhibited periodontal pathogens such as Porphyromonas gingivalis. So now we're going to talk about limitations for stem cells in periodontal regeneration. The lack of solid knowledge about the mechanisms by which mesenchymal stem cells exert their therapeutic action represents the first important limitation. Second, human periodontal ligament stem cell samples often require in vitro expansion to obtain a cell population suitable for infusion or implantation. The low number of immature cells that can be obtained by a single sample, as well as the loss of stemness when they are maintained in culture for a long time, represents a major limitation in clinical application. The protocols for cell isolation and culturing require further standardization. The large-scale expansion of mesenchymal stem cells required for cell therapies remains a great challenge till nowadays. And now stem cell biology advancements in periodontology. So recently, significant advances have been made in the field of human periodontal ligament stem cell-based therapy. From their discovery, various attempts to induce periodontal regeneration have been made by combining human periodontal ligament stem cells with scaffolds or membranes and with signaling molecules, which are molecules that transmit information between cells, such as growth hormones, cytokines, and they are very important in tissue engineering because they are essential regulators of differentiation, growth, and repair. Cell sheet technologies, which is a sheet-like aggregation of cells having an extracellular matrix, have also been developed via approaches that can involve or not the use of scaffolds aimed at supporting cell attachment, proliferation, and 3D tissue organization. An approach that consists of the direct application of autologous human periodontal ligament stem cells for intra-bony periodontal defects, bypassing ex vivo culture, contributed to defect regeneration. Ex vivo refers to the experimentation or measurements performed in or on tissues in an artificial environment outside the organism with minimal alteration of natural conditions. The advantages of stem cells for promoting periodontal regeneration would be that they have considerable therapeutic promise since they can self-renew and specialize into many distinguished cell types. They undergo immunomodulation, which plays an important role in achieving a successful outcome. And for complete regeneration of the periodontal complex, which involves not only the reconstruction of suitable alveolar bone, but also the induction of cementogenesis along the root surfaces with the oriented insertion of newly formed PDL tissue, the use of stem cells is regarded as prevalent strategy for treating periodontal disease.
Now I'm going to talk about the disadvantage of stem cells for periodontal regeneration. The anatomical complexity and low self-healing capacity of periodontal tissue make it significant therapeutic challenge to regenerate periodontal tissue. Added issues has made it difficult for them to be used in therapeutic therapy. The development of rare cancer has been linked to their implantation into human body. Next slide, please. Now I'm going to talk about the fun facts. So as you can see on the pictures, self-renewal of stem cells even after long period of inactivity. And the second one is non-dental derived cells being used for periodontal regeneration, bone marrow drives skeleton cell stem cell, and adipose tissue drives stem cell. Next slide, please. So future direction. Research is being conducted on cell reprogramming, which induce pluripotent stem cells or cells that are able to self-renew by dividing into developing into their primary group of cells. Stem cells are cells that are able to self-renew by dividing into development into three primary groups of cells. More recent regeneration techniques are being investigated include stem cells based gene therapy, modified cell pellet culture, scaffold based tissue engineering, and bioengineering tooth component assembly. Now we're going to come to the conclusion. So dental stem cells has a lot of benefit, but they also have limitations. To figure out their on cognitive potential, we need long-term clinical studies. To make teeth fully functional, research still need blood and nerve supply. Dental stem cells research has a challenge and risk. To realize the benefit, meet the challenges, and avoid the risk, stem cell research needs to be backed by effective, accountable system of socially responsible oversight and control. And this is some references. Thank you.